Hello and welcome to our very important lecture on the mechanism of action of the thyroid hormone versus the other mechanism of actions for uh, catecholamines and peptide hormones that utilize a second messenger. Now, for every rule, there is an exception. So, the thyroid hormone is not supposed to act like a lipid hormone. So, the lipid hormones, as we have discussed in the past, the lipid hormones need a lipoprotein carrier that carry that steroid lipid hormone in its core and swim with it through the blood and then cross the interendothelial gaps from the blood vessels and leave the intravascular compartment and then cross the interstitial fluid compartment and then dissolve through and traverse the bilipid membrane, the bilipid layer, because likes dissolve likes, birds of the same feather flock together. So the nonpolar substance will easily dissolve in the nonpolar bilayer leaflet of the membrane. So the steroid hormones would enter into the cytoplasm, swim through the cytoplasm, and cross the nuclear envelope, and then bind to the receptor to receptors in a mobile receptor theory, and then give us our target mRNA. The thyroid hormone is not a steroid hormone. The thyroid hormone is made up of one amino acid, tyrosine, actually two of them, two tyrosines with two iodinated tyrosines. So two tyrosines, each one has an iodine. Now, this tyrosine is a polar amino acid. It is not a lipid. It is the tyrosine that makes up the thyroid hormone is hydrophilic and lipophobic. So it is supposed to bind on the outside and then trigger an intracellular signal transduction pathway that culminates in our target response. But the problem is the thyroid hormone, although it's a catecholamine, and although it is made up of one amino acid, it acts like thyroid hormone. It acts like the steroid hormones. So the thyroid amino acid, the thyroid hormone, this tyrosine, would not need a protein carrier in the blood vessel, in the intravascular compartment. Once it is close to the target cell, it will leave the blood vessel and exit through the interendothelial gaps when the two endothelial cells undergo retraction, and then the hormone would get out, and then the hormone would cross the lipid bilayer, enter into the cytoplasm, and then from the cytoplasm it would enter into the nuclear envelope, and then it would bind a retinoid receptor. This RXR. There is an already built-in sitting receptor for the thyroid hormone. This is the thyroid hormone receptor. Thyroid hormone receptor. This THR is our thyroid hormone receptor. This thyroid hormone receptor is linked, has a very important link with a retinoid receptor, RXR. So the retinoid receptor is attached to the thyroid hormone receptor, and both of them are attached to the DNA. So all that the thyroid hormone needs is to cross into the cell, swim in the cytoplasm, cross the nuclear envelope, swim in the nucleoplasm, and then bind an already sitting, built in place thyroid hormone that also happens to be attached to an RXR, this retinoid receptor. And once the thyroid hormone binds this built sitting thyroid hormone receptor that is attached to the retinoid receptor. Now we will have a complex of an activated thyroid hormone receptor where there is a thyroid hormone that is bound to the thyroid hormone receptor, and this activation will lead to the transcription. So now we would transcribe the DNA to get our target mRNA. This is our target mRNA, and this target mRNA is going to be translated in our ribosomal subunit that is 60S and 40S, and that will give us our target protein. And this target protein will perform for us our target functions and target effects that manifest for us the effects, the clinical effects of the thyroid hormone, where we will see the basal metabolic rate going up, where we will see the temperature, or there's heat, there's increase in heat, there's an increased utilization of energy. All of these clinical effects manifest as target effects because our receptor is now activated due to the binding of the thyroid hormone receptor, leading to an mRNA, a target mRNA that will be translated in this target cell. This is our target cell. Otherwise, this is our target cell. Otherwise, our receptor will not enter except our target cell. And then that target protein will perform for us our target clinical effects. Now, this is non-steroid hormone. Obviously, we're dealing with hormones that are not steroid hormones. So the thyroid hormone, what I need you to understand is that the thyroid hormone, although it is a non-steroid hormone, it acts in a very similar manner to the thyroid, to the steroid hormones. The only difference is that the steroid hormones have receptors either in the cytoplasm, and they bind to their steroid hormone and then mobilize. That's why we say mobile receptor hypothesis. The mobile receptor hypothesis. What does the mobile receptor hypothesis say? The mobile receptor hypothesis says whenever a steroid hormone binds its target steroid hormone receptor, either in one of two locations, either in the cytoplasm, and then they mobilize to the inside of the cell, crossing the nuclear envelope, swimming in the nucleoplasm, binding into the DNA, and then transcribing for us mRNA. Or our steroid hormone would not bind to a receptor in the cytoplasm. It could bind to a receptor in the nucleoplasm, but it will never bind the receptor that is built in and sitting on its promoter region in the DNA. That is only a property of the thyroid hormone. The steroid hormone would now bind to its second alternative position, which is a steroid hormone receptor that is in the nucleoplasm and then mobilized to the promoter region of the DNA to transcribe for us. From that point on, they're all very similar, where we have a transcription of target mRNA, and the target mRNA would give us target proteins, and these target proteins are very important because the target proteins manifest for us our target responses in the cell for each particular hormone. Now, although the thyroid hormone is an amino hormone, it's made up of an amino acid, and the amino acid has an amino terminal and a carboxylic terminal, and it is charged, so that makes it polar. It makes it dissolving in water. That's why it needs no carrier in the bloodstream. It does not need a lipoprotein, but it crosses the cell, and it does not. The thyroid hormone is an exception. It does not need a second messenger. Now, other hormones, they need a separate system. Other hormones need a separate system. So now let us talk about that. Let us talk about the separate system that is now utilized, the separate system that is now utilized by the other by the other hormones that are non-steroid hormones. So typically, if you are a non-steroid hormone, you need a second messenger. You do not need a carrier because you can swim in the blood normally, but you will definitely need a second messenger because the lipid bilayer is nonpolar and lipophilic, so you cannot cross it. Therefore, we're going to need a second messenger. Now, what kind of second messenger do we need? We don't have a one universal second messenger for all of the non-steroid hormones. That does not make any sense. We have multiple different non-steroid hormones. That means these multiple different non-steroid hormones would definitely need a different second messenger because their terminal effects are different. So if a hormone has different effects terminally, different target effects, it must need a different second messenger. So let's begin with the first second messenger system. And that is the simplest and easiest of all of them. And that is the calmodulin, calmodulin second messenger system. What happens here is calcium deflects on the outside. So calcium is concentrated. It has a high concentration on the outside of the cell, but it needs to enter into the cell because there's two gradients. There is an electrochemical. There is an electrochemical gradient. Electrochemical gradient, that means on the outside of the cell, when we compare it to the inside of the cell, when we compare the outside of the cell, there is a lot of electropositive charge, a lot of calcium, electropositive charge, and here there is a little electropositive charge. So that now there is an electromotive force for the flowing of calcium from an area of a higher electropositive potential to an area of a lower electropositive potential or a higher negative potential. The other gradient is the chemical gradient. There is more atoms of calcium, more atoms of calcium found on the outside of the cell. So they need to diffuse. They have kinetic energy, they bump to each other, and now this kinetic energy forces them to move down concentration gradient. So calcium naturally wants to move in from an area of a higher concentration gradient to an area of a lower concentration gradient. But the problem is the permeability is not helping because the calcium channels are closed. Now, when the proper hormone, non-steroid hormone, binds to an extracellular receptor region, that leads to the opening. You see, now here they were closed. So calcium deflects and returns and is repelled. It does not enter down its electrochemical gradient. But when the hormone binds to the receptor, the calcium enters so smoothly, and the calcium binds to an intracellular receptor known as the calmodulin receptor. This calmodulin receptor, this calmodulin receptor is off. This calmodulin receptor is inactive. This calmodulin receptor is in the negative. So when calcium enters, because the channels are open, because the hormone bound to the receptor, calcium would now bind the calcium calmodulin receptor, and now the calcium calmodulin receptor will be activated. So now calmodulin will be from minus will become now positive, from off it will become now on, and from inactive it will now become active. And once the calcium calmodulin complex is on, once the calcium calmodulin complex is active, and once the calcium calmodulin complex is turned on, it will go and it will do some target effects inside of the cell, and we will have our target effects. And now the hormone was able to relay its message and elicit a target response in our target cell through the utilization of a second messenger. So that's an example of a one type of second messenger. Is this the only type of second messenger that we deal with? Of course not. There are multiple types of second messengers that we deal with. The second messenger system that we deal with is the G protein system. And now G protein system is very interesting. These are the G protein coupled receptor systems. G protein coupled receptor system. The G protein coupled receptor system, there is two systems that we need to be aware of. There is the G stimulatory and G inhibitory system. So the G coupled receptor protein systems are divided into two systems: System A and System B. System A is known as G stimulatory and is further divided into A1, and that is G stimulatory, and also known as A2, and there is G inhibitory. So the first system A is subdivided into A1 and A2. A1 is GS, which means G stimulatory, because it has positive effects. It will turn something on because it was off, and then it will make the enzyme active after it was inactive. That's G stimulatory. GI or G inhibitory. G inhibitory would turn a enzyme that was here. The G stimulatory turned an enzyme that is negative into positive, that is off, it turned it into on, and that is inactive, it made it active. But the G inhibitory would now do the opposite. We have an on enzyme that we need to turn it into off. We have a positive enzyme that we need to stop it, and we need to make its effects less. And we have an active enzyme, we need to make it inactive. So this is the first system. And then the second system is known as the phospholipase. Phospholipase C, G coupled protein system. So now let us begin discussing G stimulatory versus G inhibitory systems. Let us now discuss G stimulatory versus G inhibitory systems. Now, the G stimulatory versus G inhibitory systems, their cellular regulatory effects are similar. The only difference is that the target effect for G stimulatory would be to turn things on, and the target effects for a G inhibitory protein is to turn things off. So now let us begin with the G coupled protein receptor. They call it a G protein coupled receptor because it's a receptor that is coupled, that is attached, coupled and attached to a G protein, coupled and attached to a G protein. This G protein is further attached to multiple other regulatory subunits. It is attached to multiple other regulatory subunits. Now, these other subunits are known as, this is in the inactive conformation. We have GDP. GDP means guanine nucleotide that is attached to di. So there is two of them. One phosphate, guanosine GPI, and another PI, guanine diphosphate. And then it is also attached to a alpha subunit. Is attached to an alpha subunit, and a beta subunit, and a gamma subunit, When the hormone The non-steroid hormone binds to the extracellular region of the G protein coupled receptor. It will cause a conformational change. That conformational change will lead to the G protein being activated, and it will cause the GDP to hydrolyze into GTP. So we used to have guanine diphosphate. Now we will have guanine triphosphate. So we will add an extra PI, an extra phosphate group. So now we will have guanine triphosphate, and this guanine triphosphate is going to recruit the alpha subunit, and we will have GTP alpha complex. And then the other guys will be, we will now have G protein, G protein lonely with the beta and the gamma subunit. And now the GTP alpha will go and will turn on for us adenyl cyclase. And adenyl cyclase was off, but due to GTP alpha, it will be turned on. Adenyl cyclase was not working. Now adenyl cyclase is working. Adenyl cyclase was inactive. Now adenyl cyclase is active, is activated. Adenyl cyclase will go and will turn for us adenine triphosphate, ATP, adenine with 3 phosphates, 1 phosphate, 2 phosphate, 3 phosphate, and we'll turn it into adenine monophosphate. So adenine subunit with one and only one phosphate. That means we subtracted 2 phosphate groups, and this adenine monophosphate will go and will turn for us kinases that were off. We'll turn them on. Kinases, protein kinases. We'll turn the protein kinases from off to on, from minus to plus, from inactive kinases to active kinases. And we know when the protein kinases are turned on, kinases are enzymes that add plus. They add a phosphate group. So the inactive, they will phosphorylate some regulatory target. So now we have gotten to our target enzymes. So the target enzymes will now be phosphorylated due to the effects of the kinase enzyme. And now these phosphorylated target enzymes will go and they will give us target effects. And that is how we will have a G stimulatory. That's an example of a GS, G stimulatory system. A G inhibitory system is the same pathway where you would have a G protein attached to the G protein coupled receptor, but when there is an activation, the GTP alpha subunit would turn on a regulatory, in the end, a regulatory kinase, but the kinase, when it phosphorylates the target enzyme would not turn on things. It will inhibit. It will inhibit and bring down and down regulate target effect, and that is why they call it GI or G inhibitory. So that is one example. That is one example of our G protein, G inhibitory proteins and G stimulatory proteins, G inhibitory proteins and G inhibitory proteins. Now, let us look at the system of G protein that is associated, G protein system that is associated with phospholipase. Phospholipase is an enzyme, phospholipase C. Phospholipase C is a very important enzyme that is attached to the cell membrane. So when the hormone, the target hormone, the non-steroid hormone binds to the extracellular region of the G protein coupled receptor, it will cause the phospholipase C enzyme to turn from off into on, from minus into plus, from inactive into active. And that would, when phospholipase C is activated, it will snatch a fragment. It will take a bite off of the cell membrane of a very important of a very important protein known as PIP Two, phospholipid. So we would have phosphatidyl inositol 4,5 biphosphate. And this phosphatidyl inositol 4,5 biphosphate is going to be taken by phospholipase C, and it will be broken into products into two products. The first product is known as IP3, and the second product is known as DAG, diacylglycerol. IP3 stands for inositol triphosphate, and DAG, diacylglycerol is the second product. Product number one, product number two. Product number one, inositol triphosphate, is going to give us some regulatory target effects, such as increasing intercellular calcium. And when we increase intercellular calcium, that is going to affect the permeability of the cells and is going to give us some smooth muscle contractions and other target effects that are mediated by the increase in calcium concentration. Diacylglycerol is different. Diacylglycerol is going to go and activate for us protein kinase C, and that should make a lot of sense because we're dealing with phospholipase C, so the target effect is protein kinase C. And you know, when there's a kinase, we are going to be phosphorylating, adding a phosphate group to an enzyme that is off. So now the enzyme is going to be turned on because this phosphate group is going to turn this phosphorylated enzyme from off into on, from inactive into active, and from not working into working, and that will give us our target effects in question. So these are the two different systems utilized. So every non-steroid hormone will never exert its target effects except with the help of a second messenger. That's a general rule, except for the thyroid hormone, because the thyroid hormone is a non-steroid hormone, but it does not need a second messenger. So that's the only hormone that violates the rule. That's the exception to the rule. The thyroid hormone, although it's a non-steroid hormone, but it behaves like a steroid hormone, and then it performs its target effects by binding to a receptor that is built in and sitting in the promoter region. Thank you very much for your attention, and I'll see you in the next lecture.